Hi, I'm Covalent Savage. I'm doing A-level biology, and in this video we're going to be looking at immobilizing enzymes. Now, it's possible to immobilize enzymes so they can continue to catalyze enzyme-controlled reactions, but don't mix freely with substrates as they would normally do in a cell or isolated enzyme. Because in order for the product of an enzyme-controlled reaction to be generated, enzyme and substrate must be able to collide and form enzyme-substrate complexes, and this is easily achieved by mixing quantities of substrate and isolated enzyme together under suitable conditions for the enzyme to work. The product generated then needs to be extracted from the mixture, but this can be costly. So what are the different ways that you can immobilize enzymes? And, well, why do you immobilize enzymes? You immobilize enzymes because it's more efficient to use isolated enzymes to carry out reactions rather than growing like a whole organism or using an inorganic catalyst. Isolated enzymes can be produced in large quantities in commercial biotechnological processes. The extraction of an enzyme from the fermentation mixture is known as downstream processing. Now, there are many different ways to immobilize enzymes, and we're going to start off with entrapment. Entrapment is basically trapping the enzymes in a partially permeable membrane, such as a chitosan or alginate, or in a network of cellulose fibres. The enzymes are trapped in their natural state. However, reaction rates can be reduced because substrate molecules need to get through the trapping barrier. And it can sort of look, I did a tiny little diagram and kind of look like this. See, if you've got the cellulose fibres around the outside here, it traps them in the middle. Next, we can look at adsorption. Not absorption, adsorption. And this is where enzyme molecules are mixed with the immobilizing support and they bind to it due to a combination of hydrophobic interactions and ionic links. Because the bonding forces aren't particularly strong, enzymes can be detached. However, provided the enzyme molecules are held so their active site is not changed and displayed, absorption can give very high reaction rates. Next, we're going to look at cross-linking. Oh wait, no, that doesn't make sense. That does not go with this, does it? Cross-linking. Covalent bonding? Is that what my teachers called it? Okay, we're gonna, yeah. Okay. It can be called cross-linking or covalent bonding. Sorry, that really confused me. That's basically using glutaraldehyde. How do you say it? Glutaraldehyde? Or sucrose. Yeah, I love it when biologists give really easy words to say. That's probably chemists or whatever it is. Chemist? Oh, you know what I mean. Well, basically, it's when enzyme molecules are covalently bonded between the amino acids of the enzymes. This method doesn't immobilize a large quantity of enzyme, but binding is really strong, so there's very little leakage of enzyme from the support. Also, you can look at membrane separation. This is where enzymes can be physically separated from the substrate mixture by a partially permeable membrane. The enzyme solution is held at one side of the membrane, while a substrate solution is passed along the other side. Substrate molecules are small enough to pass through the membrane, so the reaction can take place. And then product molecules are small enough to pass back through the membrane. Yay! But people, oh yeah, if you want some advantages, no, I'll talk about that, so yeah. If you want advantages of immobilized enzymes, well, it makes it easy to separate enzyme from product. It also means that the product isn't contaminated with the enzyme. The enzyme can be reused, and immobilization partially protects the enzyme from the effects of temperature and pH. But, of course, there are some disadvantages. That it's expensive and time-consuming to set up. It also can be less active than free enzymes, the immobilized ones, because they can't mix freely with a substrate. So it could be a bit slower. Also, contamination is really costly. If it does occur, the whole system must be stopped and it all has to be completely started again. Yeah, so that is immobilization of enzymes. If you want to look at a gel entrapment experiment, basically this is a little diagram that we drew for my teacher. You can tell that I got an A star in GCSE art, can't you? What happens is enzymes are mixed with sodium alginate. Then drops of the enzyme/alginate mixture are then added to a calcium chloride solution. Sodium alginate forms a jelly bead with the enzymes immobilized inside. So that's basically just a little experiment with a little drawing here about it, which is so really, really lovely. But you should all be jealous of my skills. So that is Immobilizing Enzyme. There's a little video on it. I want some questions. You can compare and contrast the different methods of immobilizing enzymes. And you could also suggest and explain, because we love suggest questions, the features of bacterial cells that are most likely to be the target of the antibiotic chemicals that would be the most used to human medicine. So that is it, and good luck in your A-level biology.